Hello everyone. So in today's session we are going to discuss question related to enzyme kinetics and inhibitor from GATE Life Sciences question paper. So the question which we are having today, it is question number 3 from biochemistry section of GATE Life Sciences 2017 paper. Okay, so let's read the question what it is asking. So it is asking in an in vitro dehydrogenation reaction of succinate catalyzed by succinate dehydrogenase, malonate is added. Which one of the following curves represent the effect of malonate on catalysis of succinate dehydrogenase? Okay, so this question is from the topic of enzyme kinetics and inhibition. So before going directly to the question, let's start from some basics. Okay, so enzyme kinetics. So what happens in enzymatic reaction? So generally we have one enzyme, okay, an enzyme with an active site. Suppose this is the active site of enzyme. Active site means that's site at the enzyme where and substrate can fit, okay, like a lock and key mechanism it is. So there are two mechanisms. One is the lock and key mechanism for substrate binding, another is a fit induced mechanism. So according to lock and key mechanism, the enzyme shape is rigid, and as one key fits in the lock, similarly substrate and enzyme can fit in that way, and that's how the compatibility between enzyme and substrate comes. Other theory is the fit induced model, where when substrate and enzyme comes in proximity to each other, there are some conformational change, and then they get bind to each other, and the reaction will carry forward. So these are both the theories, but basically there is an active site where the substrate will come and bind to the enzyme. So suppose this is my active site, and I have a substrate. This is my substrate which can go and bind to this enzyme. Okay, this is how. Okay, so this is called an enzyme substrate complex. Okay, it is a transition state. It is in a transition state. Okay, so we have a free enzyme, we have a substrate, it binds, enzyme substrate complex, which is a very high energy transition state, and after this step the enzyme will free enzyme is released, and now this substrate is getting converted to product, different structure of product, and at the end of the reaction we get our free enzyme back. So this is what happens in a basic enzyme kinetics, and Michaelis-Menten have plotted this, which is called Michaelis-Menten graph, velocity, substrate. Okay, so as you start increasing the substrate concentration, velocity of your reaction will also increase. Okay, but it is not a linear graph, it is such how hyperbolic curve. Okay, so at some point you'll start seeing saturation. So this is the point where maximum velocity has been achieved. So this is called Vmax of the reaction. Okay, and if this is the Vmax, half of the Vmax, half of the Vmax, if it is here, so the concentration where the reaction reach to the half of the maximum velocity, that particular substrate concentration is called as Km, which is Michaelis-Menten constant, or Km. Okay, so from this particular graph, Michaelis-Mentens have derived one formula, which is okay. So here, if Here if Km is equal to S, then if Km is equal to S, then V is equal to 1 by V max. V is equal to 1 by V max. So this is the equation we get generally from this. Okay, so Km is the velocity, Km is the substrate concentration at which rate of reaction is half of the maximum velocity. But plotting the reaction kinetics or Bmax or demonstrating Vmax using a hyperbolic curve is quite difficult, because at very higher substrate concentration we generally achieve the Vmax. So plotting Vmax or finding out the value of Vmax using a hyperbolic curve was quite difficult. So to avoid that, this Michaelis-Menten curve is converted to Lineweaver-Burke plot. Lineweaver Lineweaver Burke plot, where they have taken the double reciprocal values, so Vmax as well as the substrate. So these values were reciprocated and plotted in such a way that now it gives a hyperbolic curve. So reciprocal of substrate and reciprocal of velocity, and using this we generally get a linear line. So using this linear curve, now it is very easy to estimate the Vmax. Okay, so here we are getting two intercepts, so on y-axis and x-axis. So y-axis we are getting one intercept that is called, that is 1 by Vmax. On x-axis what intercept we are getting, that is our 1 by Km. Okay, we can say minus 1 by Km because it is in the negative on this ends, and the slope of this line is Km by Vmax. So Michaelis-Menten curve is converted to Lineweaver-Burke plot by using the double reciprocal values of velocity and substrate to convert into a straight line, so that we can easily find out the value of Vmax. And this plot is very useful to show about the inhibitor's kinetics as well. So this is the plot, Lineweaver plot, which we are going to follow. Okay, so before jumping to that, let's first discuss about the type of inhibitions. What are the different types of inhibitions? Okay, so types of inhibitors. So as written in enzymatic reaction, it can be inhibited by several chemicals as well. So in that inhibitor category, we have generally two types with two broad categories. One is the irreversible inhibitor. Another is the reversible inhibitors. So irreversible inhibitors are some kind of permanent inhibitors, we can say, which damages the active site of enzyme, or you can say destroys the functional group of enzyme, destroys the functional group of enzyme which is important for its activity. Okay, and yeah, which is important for its activity. So it makes a permanent kind of damage on the enzyme, and that cannot be reversed back. Okay, so this is a very strong kind of inhibition, which is irreversible inhibitor. Okay, example of this I would you must have heard about cyanide inhibitor. Cyanide inhibitor or cyanide as a uh, very toxic chemical for human consumption. So cyanide, what does it do? It uh, binds to cytochrome oxidase enzyme in an irreversible manner. Okay, and cytochrome oxidase, which is involved in mitochondrial etc, and mitochondrial etc which produces ATP. So without when the cyanide inhibits the complex 4 or the cytochrome oxidase, it uh, hampers the mitochondrial etc, which hampers the production of ATP. And as we all know, ATP is required for all the physiological function. So in absence of ATP, all the physiological processes are inhibited, and it is highly lethal for human consumption. So this is a great example of irreversible inhibitor. Other than that, we have an example of inhibitor, DPF. Okay, so this DPF, this is an enzyme which is having a functional group, as I'm telling you that it destroy the functional group. So as it is having a functional group, this is the functional group of enzyme, and its substrate, which is it inhibits generally acetylcholine esterase. So if it has a substrate, so this is the substrate. So this lone pair, so this is an inhibitor. Okay, so this reaction, so this fluorine, so this attack on this lone pair, and further yeah. So this is the enzyme, and this is the inhibitor of this enzyme. So this is the functional group of enzyme. So this inhibitor with this fluorine attacks on the enzyme and forms a covalent bond. It has formed a covalent bond now. And now, as you can see, because of the covalent bond formation, the functional site of this enzyme, functional group of this enzyme is lost, and it is has lost its enzymatic activity. Okay, so this is also one of the example of enzyme irreversible inhibitor. Okay, so other than that, we have suicidal inhibition in the reversible inhibitor category. So suicide inhibitors are the specialized kind of irreversible inhibitor, which is also called as mechanism based inhibitor. Okay, suicide inhibitors, or we can say them mechanism based inhibitor. So these inhibitors, what they do, they utilize the machinery of enzyme's own mechanism to convert the substrate into product. So they bind to the enzyme, convert the inhibitor in a more active or toxic inhibitor that completely destroys the enzyme and let the enzyme suicide, leaving it non-functional. So that is why it is called a suicide inhibitors. Example of suicide inhibitor is allopurinol. This binds with the enzyme and converts into a further toxic, very strong uh, molecule, which is oxopurinol, and which is now uses the enzyme's own mechanism based action, converts into a more toxic product, and destroys the enzyme. So this is a example of suicide inhibition. Now let's talk about the reversible inhibitors. So reversible inhibitors are the inhibitors which doesn't cause a permanent damage on the enzyme activity. This uh, type of inhibition can be reversed as well. Okay, so in reversible inhibitor we have three types. One is the competitive inhibition, another one is the uncompetitive inhibition, third one is non-competitive inhibition. Okay, so competitive inhibitor is very simple. So as we have discussed, like an enzyme has an active site where substrate can bind. So this is, if this is our substrate, okay, so inhibitor could also be structurally similar to the substrate. Okay, this is my substrate, this is my inhibitor. So substrate inhibitor could be structurally similar. And that is the key point in competitive inhibition. Structural similarity between enzyme and between inhibitor and substrate. Okay, so they both can bind to the active site of the enzyme. So that's why there is a competition between the substrate and the inhibitor to bind to the active site of the enzyme, and whichever is having in a higher ratio, that will overcome the binding. So if we have higher inhibitor, lesser substrate, then more inhibitory reaction will take place. If we have more substrate, less inhibitor, so it can be replaced. So here we get an enzyme substrate complex where we'll get enzyme plus product. In this case also we get an enzyme substrate enzyme inhibitor complex, but this cannot be converted into product. This is a reversible reaction. Yeah. So this is what happens in the competitive inhibition, as they are competing for the same uh, site. So what happens? As we know, the Km of the reaction we already discussed is the concentration of substrate required to reach half of the Vmax. Okay, so higher the Km, lower the binding capacity or lower affinity. So here also, in presence of inhibitor, because it is the structural analog of substrate, the affinity of substrate towards the enzyme becomes lower. That's why the Km value of in this reaction, it gets increased. Okay, Km value gets increased in competitive inhibition. Now next we have uncompetitive inhibition. Uncompetitive inhibition is uh, one where here we shown that competitive inhibitor bind to the free enzyme, but in case of uncompetitive inhibition, this inhibitor is binding this inhibitor is binding to enzyme substrate complex. This is my inhibitor, this is my substrate, this is my enzyme. So this inhibitor is binding to enzyme substrate complex. So this uncompetitive inhibition generally takes place in those enzymes where 2, 3 or more substrates are involved in some enzymatic reaction. So there these kind of inhibition happens. So what it does that it impacts enzyme in such a way, because it is only binding to the substrate bound enzyme, so in somehow decreases the Km of the reaction, and also the Vmax of this reaction is also changed. But in this case only Km gets increased, but the Vmax of the reaction remains the same in case of inhibitor as well as in the substrate. But here in this case, where the Km and the Vmax both get decreased. In non-competitive inhibition, so here non-competitive inhibitor can bind to the free state of enzyme as well as the enzyme substrate complex of the enzyme, because non-competitive inhibition bind to the allosteric site other than the active site, allosteric site of the inhibitor. Okay, so it is binding to the allosteric site of the inhibitor. So what is happening, because it is binding to the allosteric site, the active site is remain unhampered. That is why there is no change in the Km, no change in the Km, but the Vmax in this reaction gets decreased. Vmax decrease in the non-competitive inhibition. All these three can be shown in the Lineweaver Burke plots for enzyme inhibition. So in the first case, as we have seen, okay, so here in the values are changed by a constant which we are calling as alpha. Okay, so this is my uh, blue one is a graph without inhibitor. This is a competitive inhibition graph. So here I have already mentioned that the Km increases. Okay, the Km increases here. So the new Km will be, so here it increases by alpha Km, but the Vmax remains the same, no change. Okay, in this case we have both Km as well Vmax, both are getting decreased by alpha. In this third case, non-competitive inhibition, our Km is getting decreased, the new Km, Vmax, and Vmax, no, here the Km remains the same because a non-competitive inhibitor binds to the allosteric site, not the active site, but Vmax decreases. So these are the short formulas we have to follow for our Lineweaver-Burke plot for enzyme inhibition. Okay, so if we'll see our question, so our question states that in an in vitro dehydrogenation reaction of succinate catalyzed by succinate dehydrogenase, malonate is added. So if I talk about the structures first, if you know the structure, you can easily come back to the conclusion. So this enzyme is succinate dehydrogenase. The substrate is succinate. For this enzyme, the substrate is succinate. So if I'll draw the structure of succinate, so this is my succinate. Okay, so these bind to the enzyme, convert it to another, after dehydrogenation, this is the product which we are getting, which is fumarate. Okay, and the enzyme gets free. This is the free enzyme, product plus substrate. This is enzyme plus substrate complex. Okay, so in the same we have added the malonate. So malonate structure is okay. So if we'll see the succinate and malonate structure, malonate and succinate are structurally similar, and both these carboxylic acid residues are present in succinate and malonate, which can fit into the active site of the enzyme. So in place of succinate, malonate can also go and fit, but as we have seen here, And as we seen here, substrate can only make the product, not the inhibitor. So even though the fumarate will malonate will go and bind, but it will not make any product. Okay. So because of the structural similarity, malonate can also fit in the enzyme succinate dehydrogenase, but it will not form any product. So that is an example of competitive inhibition. So here it's asked to us that which one of the following curves represent the effect of malonate on the catalysis of succinate dehydrogenase. So as it is competing for the same binding site or the active site, so that means it will affect the Km of the enzyme kinetics. So Km will be increased in this case because the affinity of succinate will decrease in presence of malonate, but the rate of reaction or the maximum velocity will not hamper. So here at the y-intercept we know that we have the Vmax, and s-intercept we know that we have the Km. So if we'll see in all four graphs, in all four the y-intercept is changed, that is the Vmax is changed, but according to our theory the Vmax will remain the same. That is why we'll go with the first option. And here also if we'll see this, our Vmax is remaining the constant, but the Km is changing. So if Km is increasing, so it is without inhibitor plus inhibitor, so here the Km is will increase. So according to the Lineweaver plot and competitive inhibition kinetics, our correct option will be option A. This is my reference, NPTEL reference for this lecture. Thank you.